Welcome to the Kansas Biological Survey Friday Ecology Seminars. My name is Dan Reuman. This semester we're having intermittent alternating hosting between myself and Sarah Baer because the usual host, Ben Sikes, is on sabbatical, but he'll be back next semester. So my basically whole role here is just to introduce Jasmin and then to sort of help manage the questions at the end. So I'll get started introducing Jasmin. So Jasmin, this is Jasmin Albert. She graduated from an undergrad at KU in May. Prior to that, she had been working, and since then, she's been working in my lab as a researcher, an undergraduate researcher, and then a post-baccalaureate researcher. She will be going to grad school next year. She's been accepted a few places, but hasn't decided officially where she's going to go yet. So I just want to point out how impressive that is. Jasmin is doing what you'll find out is very good and high-level research here as an undergrad and leading up to going to grad school. And she brings together an impressive collection of skills in ecological insight, together with mathematical and statistical and computing abilities, all of which should be demonstrated in this talk. And with that, I will let her take it from here. Go ahead and share your screen, Jasmin.
Thank you, Dan, for that very nice introduction. Thank you for everyone for being here at my talk. Okay, do you guys see the presentation? Yes?
Yes.
Okay. All right, so my talk is called Asymmetric Relationships and Their Influence on Competition and Coexistence. And like Dan mentioned, I've been working on this with him for like a couple years now. Okay, so in this talk, I'll first go over what I mean by asymmetric relationships. I'll give a brief review of coexistence theory. I'll talk about how these two things connect. And then at the end, I'll give you my results of applying these ideas together to a couple systems. All right, what the heck do I mean by asymmetric relationships? So first, let's think about any two variables that might have a relationship. It can be rainfall and plant diversity, hand washing and germ spreading, or exercise and muscle size. If we plot these two variables against each other, one on the y-axis and one on the x, what might that look like? Well, if these variables are highly correlated, we might think of something like this, right? So this is a nice symmetric bivariate distribution. Well, what if the relationship looked like this? These two variables are still very much correlated, but the structure or the pattern of the relationship is very different. So if we look closer at this purple distribution, we can see that in the right tail, the variables are very related to each other, and down here in the left tail, they're not as related to each other. So this is what I mean by asymmetric relationships. One tail of the distribution is more related than the other tail. And this is what we call a right tail associated asymmetry, and it can go the other way too. So this is a left tail associated asymmetry. And here's all three of them together. They're pretty different, and we can describe these asymmetric relationships as having asymmetric tail associations, or what I will call ATAs, and I'll use this acronym for the rest of the talk. Okay, so what if I told you that if you actually measured the correlation of all three of these relationships, that the correlation value would be very, very similar? Would you believe me if I told you that? Okay, well, it doesn't matter if you believe me or not, because it's true. These three bivariate distributions have strikingly different patterns of association. Some of these are asymmetric, and one of these is perfectly symmetrically associated. However, despite these differences, they all have the same correlation value. Pretty crazy. Okay, so why does this all matter? Personally, I think it's pretty deceiving of correlation to blindly generalize these three different distributions like that, but let me give you an example. Okay, so let's say you're doing research on limiting nutrients of plant growth. You go out into the field and let's say you measure plant growth over time and you also measure the concentration of nitrogen, phosphorus, and potassium. Then you load your data into R and let's pretend that R tells you that all the nutrients are correlated with plant growth to the same degree. However, the plots look like this. So which one is actually the limiting nutrient? Well, a limiting nutrient limits a plant's growth when that nutrient is rare, right? So the answer would be nitrogen, because the nitrogen concentration when it's small is more related to the plant's growth when that is small as well. So growth is more closely related to nitrogen when nitrogen concentration is small. So then we can say if a plant's growth shows left tail association with a nutrient, then that nutrient is likely to be limiting. To give another example, we can think about spring monthly rainfall. So generally, March and April rainfall is probably correlated. When March is dry, April is also dry, and when March is wet, April is also wet. But of course, there's going to be some years where this isn't true. So what if in a particular location, there's very intense, long-lasting floods that happened almost every year? Well, then these very high values of precipitation would be more correlated than in the lower values. So then we would see a right tail association between the relationship of March rainfall and April rainfall. And that would look something like this. To give an example outside of ecology, in finance, left tail associations between the success of one stock and the whole market can be used as risk assessment for predicting a crash. So if the relationship between one stock value and the whole market is left tail associated, then the two are more likely to crash than boom together. And then that stock can be used as a predictor of whole market crash. Okay, so I'm giving all these hypothetical ecological examples, but are they even true and why should we care? Well, thanks to research in the lab, ATAs have been found to be common in nature. Turns out most things, when you plot them against each other, don't really have a perfectly symmetric relationship. And some examples of this include right tail association between soil carbon and soil nitrogen, and also a right tail association between species metabolic rate and their biomass. So not only are they common, but they've also been shown to be important for certain ecological phenomena. For example, extinction risk can be accentuated when environmental fluctuations are spatially more left tail associated. So what do I mean by that? How can asymmetric tail associations be important for extinction risk? So let's suppose that there's these three populations of the same species separated in space. And the metapopulation mitigates extinction risk through dispersion. So when one population is near extinction, it can be rescued from extinction via incoming individuals from the other populations. So the claim is that when the population's environmental responses are left tail associated, the extinction risk of the whole metapopulation then increases. What that means is that when all the populations are in the lows at the same time, there's no rescue effect of dispersion. You can imagine a mechanism of this might be a large climatic event such as a large drought that affects all three populations at the same time, and that causes them to crash all at the same time. On the contrary, when there's not a large climatic event and weather fluctuates at the smaller spatial scale, the populations fluctuate normally and asynchronously, and the metapopulation persists. Okay, so ATAs are common in nature. They're important for some parts of ecology. So now we want to know if ATAs can be important for a different part of ecology, coexistence and the maintenance of biodiversity. So it's long been questioned, how do species that compete for the same limiting resource coexist? And according to the competitive exclusion principle, if species depend on the same resources, eventually the strongest of those species would drive the other species to exclusion through competition. Well, it turns out that environmental fluctuations can be crucial to the coexistence of species and the maintenance of biodiversity. Just like the coexistence of these warbler species that depends on resource partitioning in the space of the trees, species can depend on different partitions of time when the environmental conditions are ideal for them. And this is known as temporal niche partitioning. To give an example of this, here's a population of a species over time. So let's call this species 1. And species 1 increases in abundance when its environmental conditions are favorable, and then it decreases when the environment is bad. And let's suppose that when the environment is bad for species 1, it's good for its competitor, species 2. So that's here in the red. So again, when one species is high, the other is low because they respond differently to the environment. And although one species may become rare, it never goes extinct. So in this case, coexistence means that this species can increase when rare. And this growth rate when rare is the metric that we use to quantify the coexistence of species. Okay, so let's break down this growth when rare. So according to modern coexistence theory, growth when rare can be attributed to a few mechanisms. We have the contributions of coexistence from constant conditions, contributions from variation in the environment, delta E, variation in competitive pressure, delta C, and then we have this variation in environment and competition together, which is represented by this delta E sharp C and delta EC, but let's not worry about this first sharp term right now. And some might be familiar with this delta EC term. It's called the storage effect. So when you add all these terms together, you get the growth rate when rare. And the sum of this answers exactly that. Can the species increase when rare? And if the sum is more than 0, then the species can persist. And in order for coexistence to occur, all species in question must be able to increase when rare. Okay, so what about ATAs? How will they fit into coexistence? So like I mentioned, growth rate when rare decomposes into various mechanisms. Again, we have contributions from constant environment, environmental variation, variation in competition, this weird term that we're not going to worry about, and finally the storage effect. And to investigate ATAs, we are interested in the storage effect. Okay, so why must we study the storage effect to investigate ATAs? The simplest way to answer is because quantitatively, the storage effect is the contribution to coexistence from covariation in the environment and competition. And if you recall from earlier when I was introducing ATAs, covariance is a pretty limited descriptor of relationships because it doesn't provide any information about any possible asymmetry between the two variables that there might be. So we can go over the storage effect a little bit. So again, storage effect is this measure of EC covariance, where E is a measure of how good the environment is, and C is a measure of how much the presence of a competitor hurts a species. So essentially, it indirectly helps the rare species by hurting the growth of the common species. Because the common species is abundant, a good environment that is positively associated with high competition will hinder the common species from taking advantage of the good environment, and then the rare species can increase with low competition. So to summarize, storage effects and EC covariance benefit the rare species by limiting the growth of the common species when the common species is in its good environment. Okay, so now that we're all on the same page, we know that coexistence can be decomposed into different mechanisms. We know that storage effect is one of those mechanisms, and it's defined by EC covariance. We also know that covariance doesn't tell us very much about asymmetric tail associations, and that would suggest that E and C can covary in any pattern, whether asymmetric or not, and the contribution of storage effects to coexistence would be the same. So that raises the question of, can we decompose the storage effect to tell us about the importance of ATAs for coexistence? And do ATAs play a meaningful role for coexistence and the maintenance of biodiversity? Okay, so to answer that first question, it involved a lot of math, and the mathematical details are way too exciting to include in this talk, so I'm just going to skip over to that. And the answer is yes, storage effect can be further decomposed. And we decomposed the storage effect into these two terms, which we call the contribution of ATAs and the contribution of correlation per se. And essentially, we quantify the contribution of ATAs by computing the storage effect as you normally would, and then we subtract out what the storage effect would be if E and C were perfectly symmetrically correlated. So now that we have this math figured out, we can move on to the next question where we use this math to figure out if ATAs have a meaningful contribution to coexistence. Okay, so we applied our decomposition to find out if ATAs are meaningful to coexistence. And we first explored this using a theoretical model, and then we moved on to a real system using a competitive diatom model. So again, the math is way too exciting to include in this talk, so I'm skipping to the results of the theoretical application. We found that ATA contributions were meaningful, and sometimes ATAs can even play a driving role in species coexistence, meaning that when there were no ATAs, coexistence was not possible. And I called this ATA rescue. On the other hand, we also found that the presence of ATAs can drive populations to exclusion, meaning that sometimes when there were no ATAs, coexistence would be possible. And I called this ATA exclusion. So here's an example of ATA exclusion. On the X, we have time, and on the Y is the population of a species. So on the top is if environment and competition were perfectly symmetrically related, and on the bottom, we have a simulation where E and C were asymmetrically associated. And we can clearly see that over time, when E and C are asymmetrically associated, the species becomes excluded and coexistence is not possible. So to summarize this, in our theoretical application using simulations, we found that ATAs were non-negligible, meaningful, and sometimes crucial for coexistence. Okay, so moving on to our empirical application. We chose to investigate a previously studied diatom system by Descamps, Julian, and Gonzalez, published in the journal Ecology. And in summary, they grew batches of 2 diatom species, both alone and then in competition, in constant temperature and in a fluctuating temperature. They found that constant temperature led to the exclusion of one species by another, and that fluctuating environmental temperature led to the coexistence of the 2 species. And they concluded their study by suggesting that the storage effect was likely a driving mechanism of the diatom's coexistence. And so now we want to use this example to mathematically decompose their coexistence and ask if ATAs were meaningful. Fortunately, a lot of work has already been done for providing a framework to decompose the coexistence of the diatoms. And we have these 2 ecology letter papers to thank for that by Stephen Elner and colleagues. So in order to decompose the coexistence and EC covariance, we need to define what E and C are. So here, E is defined as the temperature fluctuation defined by this sine wave with an amplitude of 6 degrees, mean of 18 degrees Celsius, and period of 60 days. And we define C, competition, as this ratio between nutrient uptake and available nutrient concentration. So we were interested in the coexistence and ATA contribution beyond just this one definition of temperature fluctuation. So we simulated and decomposed diatom coexistence at varying means, amplitudes, and periods. Okay, so moving on to results. For the results, I'm going to show different plots of coexistence mechanisms, including our ATA contribution and correlation per se, against different parameters of temperature fluctuation, the amplitude, period, and mean temperature. And just as a refresher, these are some of the equations. So this first one was the original decomposition with the storage effect on the end here. And then what we did was decompose the storage effect into these two terms. We have the ATA contribution and the correlation per se. And then when you put those two together, you get this last equation here. Alrighty, so how does amplitude of the temperature fluctuation affect coexistence and ATA contributions? So we have this plot here. We'll have the coexistence mechanisms plotted on the y-axis and then the amplitude of the temperature on the x. So here's my legend, and we'll go ahead and plot these other mechanisms that we don't really care about, but generally most of them increase as the amplitude of temperature fluctuation increases. And then let's plot the ATA contribution and correlation per se. So here, ATA contribution is in red and correlation per se in blue. And you can imagine if you add these two together, that would be the storage effect. So this answers our question of whether ATAs are important for coexistence. We can see that the ATA contribution is comparable to the other mechanisms. So it's just as important as the other mechanisms in maintaining coexistence. So now we plot the sum, the growth when rare. And like most mechanisms, it increased with amplitude and, but when the amplitude was small, the growth rate when rare was negative, meaning that coexistence is not possible there. And that makes sense because when amplitude is small, the temperature becomes more similar to constant. And in the actual experiment, when temperature was constant, coexistence was not possible. So if you look closely at where this orange line and the red line intersect, you can see that there's a small space where the growth rate is larger than 0, but smaller than the ATA contribution. And this is where ATA rescue happens, because if the ATA contribution was not there, then the growth rate would be negative in this small little space. Okay, so moving on, how does the periodicity of the temperature fluctuation affect coexistence and ATA contributions? So similarly, we have the same plot setup. I'm just going to go ahead and plot all the things at the same time. Okay, so here the ATA contribution and growth when rare is here. And we see that ATA contribution is comparable to the other mechanisms in black. But in general, changing the period of the temperature fluctuation doesn't really change the contribution to coexistence of these other mechanisms, as well as the total growth when rare. So moving on to the mean temperature of the fluctuation, the mean of the temperature fluctuation, same plot setup. We have the other mechanisms that we don't care about here. They, like amplitude, they increase with the mean of the temperature fluctuation. And here we have the ATA contribution and the growth rate when rare. And similarly, the ATA contribution is comparable to the other mechanisms, so it's just as important. And here we actually have a wider band of ATA rescue where the growth when rare is positive, but less than the ATA contribution. So if the ATAs weren't there, then coexistence would not be possible. So essentially, the results of this diatom investigation show that ATAs can contribute meaningfully to the coexistence of diatoms. And in some cases, ATA rescue occurs where if there's no ATA contribution, then coexistence would not be possible. And in conclusion, in this research, we set out to mathematically quantify the contributions of ATAs to coexistence, and we did that by providing a formal extension to the decomposition of growth when rare. And 2, we used our decomposition of storage effects to find that ATAs are non-negligible and contribute meaningfully to coexistence, and in some cases are the driving mechanism of coexistence or exclusion. And to reiterate some highlights of this research, it highlights the fact that common measures of relatedness, such as correlation or covariance, don't really tell you the whole story about the relationships between variables. And it highlights the importance of giving consideration to asymmetric tail associations in ecology, and adds coexistence theory to the list of ecological questions that ATAs have been found to be important for. And there's many avenues to be further explored with this research. When one could continue the exploration of the previously studied systems to for a meta-analysis of sorts, or you could do like field experiments to detect ATAs between species interactions in their environment and to decompose their coexistence fully using our decomposition. And it would be interesting to look into how ATAs might be changing with climate change and extreme climatic events. And our research kind of suggests that ATA specialists might be a thing where species can exist where ATAs are present, but cannot exist when there are no ATAs. So with that being said, I'd like to thank everyone for coming to my talk, and thank you for all the lab members for all the helpful discussions and helping me disseminate my research. And thank you to Dan for supporting me through this research.
Great. Thank you very much. I see lots of little applause icons appearing. That's great. Thanks a lot, Jasmin, for a very informative talk and entertaining and interesting. Now is the time for questions. So Jasmin, you may find that you need to reshare your screen to answer the questions, but I think the way to do questions is for people to just kind of indicate that they want to speak and or unmute yourself and start talking in this sort of orderly way, and then we'll take it one question at a time. Go ahead.
So Jasmin, that was a great talk. So early on, you identify that the difference between right and left tail association with your ATA, but then when you started doing your modeling, you sort of dropped the distinction. Were you always imposing right tail association with ATA in your theoretical consideration and your simulation, or were you having it be equally likely that it's left or right associated?
Yeah, thanks for your question. So in our preliminary investigations, we found that it doesn't make a difference if they were right tail associated or left tail associated.
That was for the lottery model.
Yeah, in the theoretical application. But yeah, I think in the math, it turned out that there was no difference if they were left or right tail associated.
That's interesting, isn't it? Because you started out identifying that, at least for the limiting nutrient expectation, that if it was what left tail associated, you'd expect it to be more important for coexistence because it is then that environmental variable is more associated with growth when it's rare. But that ends up not mattering when you actually look at the lottery model.
Yeah, that's in the lottery model, which is a pretty simple model. So you can expect that to be true in other cases.
But what did you do with the algal model, with the diatom model, which really is very much like the limiting, it's the classic limiting nutrient system. Even in that system, it didn't matter whether it was right or left tail associated?
Well, what we did there was, so the asymmetric association was like already there in the data, right? So we just separate out that asymmetry, and then, yeah, so we have the storage effect, and then we subtract out what the storage effect would be if it was perfectly symmetric.
So which direction was the asymmetry in the diatom data?
That one, the E and C were negatively correlated, and they were more correlated in the upper tails.
Which is counter to your expectation, like for a limiting resource, which is interesting, isn't it? That's still, so if it was the other way, that like that range where the tail association mattered might expand, maybe? Is it, that's in that system?
What do you,
I guess
sorry.
you can't really guess that because the data is what it is. It's really cool, Jasmin.
Yeah, thanks for your question.
I can't see everyone on my screen at the same time. So if I'm not calling on you, just unmute yourself and ask away. I see Nat with a hand raised. You want to talk, Nat?
Yeah, Jasmine, great talk. Uh, Throughout this, some of the framing has been focused on the left-tail association, as Professor Beaver mentioned. Um, if you were to switch and go to right-tail association, could you think of any cases in the literature where there's, just because you've definitely read more about coexistence than I have and know more of the case studies people have done, cases where right-tail associations could drive stochastic exclusion events? Given that MCT is like stability through time to some degree, and the ability to maintain coexistence, can you think of cases where right tail associations and accounting for it might actually improve the odds of detecting cases where coexistence isn't possible?
I think I would have to go back to my example of the rainfall. So like in that case, if you have extreme drought events, you would have right tail association between month to month rainfall. And that could go the other way too if you look at drought and like low rainfall. So yeah, maybe like extreme rainfall events could affect coexistence in that way, if that answers your question.
It was more of a, could the common species be right tail associated and thus outcompete even in good years? And have spike events that pressure out the rare species. And if not, that's fine. Like, I just wanted to know if you'd encountered anything like that in the literature.
Yeah, no, I'd have to think more about your question to answer that.
Thank you.
Thanks.
Any more questions? I've got one. Have you thought how you might, the diatom example, as far as I understand, based on what you said, is sort of, the experimental work was done back in 2005. So there was no actual manipulation of ATAs. It's like manipulating a model based on the experiment, right? So I guess my question is, have you thought about how you might use the diatom system or another system to actually do an experiment which would kind of manipulate ATAs and show hopefully differences in coexistence in different treatments, which might correspond to different ATA setups? Like, is there a good attractive, do you have any thoughts on it? What would be a good system for like actually bringing the experiment closer to the result?
Yeah, so going back to the rainfall example, I think an application of it could be where you manipulate rainfall to get very intense rainfall over a long period of time. And that would be your case where you have ATAs and then just have like normal ambient precipitation as your control. And I hope I can do that when I go to grad school.
So I had a question, sort of a much more basic question. Is there a simple measure of the degree to which you have ATA? I'm thinking of, if you've got a data set or correlation you're interested at what point do you have to start thinking about ATAs as a factor? Is there, how do we kind of look at that threshold if there is such?
Do you mean like how you would measure if there are ATAs present?
Yeah.
Yeah, so Dan actually came up with this, but you would basically measure the correlation in like one half of the distribution and then the correlation in the other half, and then you would subtract those. And if they're uneven, then yeah, then you would have ATAs.
Thank you.
Yeah, thanks for your question.
There's no way to know. It would be system specific, I would think, like how your TAs have to be in order to produce a biological effect, right? Like it may be that you need very extreme asymmetry of tail associations to produce an effect, or it may be that just a little bit of asymmetry in tail associations will produce an effect. And I would think that would depend on the system. Okay, if there's no other questions, I think we should thank Jasmin again for a great talk by demonstrating with our hands or our little icons. And thank you all for joining us, and see you next week.
Thanks, everyone.